I'm Antony Dodd. I'm based at the John Innes Centre, which is just outside Norwich. What I'd like to do today is I'll start by giving you a kind of potted history of my career. Then I'll give you some general background to the discipline or the field that I work in. And then after that, I'll show you two snapshots of pieces of research that we've done recently in the group that you might find interesting. So I did my undergraduate degree at the University of Newcastle, or University of Newcastle upon Tyne it was called back then. And I did my PhD also at Newcastle University with it's supervised by Professor Anne Borland and Professor Howard Griffiths. And then after that I moved to Cambridge to Professor Alex Webb's lab where I worked as a postdoctoral researcher. And then in a moment of insanity, the Royal Society awarded me a university research fellowship to establish my own research group, which I took to the University of York, just down the road from here. And then I got my first permanent academic position in Bristol and moved to the John Innes Centre a couple of years ago, where I'm currently also head of Department of Cell and Developmental Biology. I think that some of you will have heard of the John Innes Centre but might not know very much about it. The John Innes Centre is a centre for excellence in research in plant and microbial sciences, and we have a mixed economy of very fundamental research through translational research to also commercial endeavours based within the Institute. And the Institute receives funding from a variety of different sources. And our current overarching goals are to contribute to feeding the world, combating health threats, and contributing to net zero through tackling climate change. So that's the work of the Institute overall. And we're about 400 people at the Institute, of whom about 100 are PhD students, and we also have some undergraduate summer interns who come and join us at the Institute, and for example year in industry students as well. So it's not something that you can't engage with if it interests you. So let's now focus in a little bit on the work that my research group does. So you'll know that the Earth rotates on its axis, and this creates cycles of day and night due to the position of the surface of the Earth relative to the Sun. And these changes in the position of the Earth create on the ground daily 24-hour cycles in the presence of light and dark, and they cause daily temperature fluctuations as well. And what these changes do is that they impose a variety of selection pressures on the life on the surface of the Earth due to the availability of light for photosynthesis for plants, or due to the behaviour of pathogens, or the daily fluctuations in the relative humidity. And what this means is that plants growing outside, including all of our crops, experience 24-hour changes in environmental conditions. And what these 24-hour changes have done is they've selected for the evolution of something that we call circadian clocks. Okay? Now, so the word circadian comes from about a day, and circadian rhythms or circadian clocks produce a cellular or biological measure of the time of day that allows organisms to anticipate and respond to the 24-hour cycles in the environmental conditions that exist outside. So you have one if you travel through time zones, if you travel long haul, you are aware you have a circadian clock because the phase of your clock becomes misaligned from the time in the environment, and you experience that as jet lag. And just like we have biological clocks other organisms as well on the planet, including plants, have biological clocks. Firstly, I'll introduce to you what a circadian rhythm is. There's a kind of formal definition of it. So a circadian rhythm is a biological cycle that has a period of about 24 hours, as suggested by its name. And what this means, so in this diagram here on the slide, the first part of the diagram is supposed to illustrate a day and night cycle where the yellow blocks represent the daytime and the blue blocks represent the nighttime. And under these conditions, this wavy yellow sine wave represents a physiological process, just an arbitrary physiological process. So under the cycles of day and night, this process is rhythmic. But then upon transfer to, for example, conditions of constant light and constant temperature, that rhythm will persist, and it will persist with a period of about 24 hours, and it will occur in the absence of external cues. So we call this circadian free-running conditions. So the rhythm is generated from within the cell. And this self-sustaining nature of the biological rhythm occurs because the rhythm arises from what is called a biological clock or a circadian clock. And this is we'll come on to that in a minute. Another feature of circadian rhythms is a relatively unusual property in biology. So you know lots of things like the activity of enzymes, for example, depends on the temperature. And as you increase the temperature, the enzyme activity increases. But an unusual property of a lot of circadian rhythms is that they have what we call temperature compensation of the circadian period. So across a range of physiologically relevant temperatures that the organism might encounter in nature, for example, between 10 and 30 degrees in Arabidopsis plants, the period of the rhythm remains relatively stable at about 24 hours. So it has a kind of internal buffering that provides resilience to temperature changes. So it's a relatively unusual property. So what this means is that the circadian clock remains accurate across a range of different temperature conditions. And another thing to remember about circadian rhythms is that they're found right across life. So yes, we're talking here today about plants, but circadian rhythms are found in a variety of different types of organisms. There's been recent reports of circadian rhythms in bacteria, through photosynthetic bacteria such as cyanobacteria, fungi, algae, multicellular algae, and through to vascular plants and various kinds of animals as well. So they're pervasive throughout life, and they're thought to perhaps have evolved independently on a number of different occasions. We know that from the kind of molecular genetics of circadian clock structure. It's the case also that people that work with circadian rhythms or study them, which we might refer to as chronobiologists, often select different experimental models for their experiments depending on the type of question that they want to answer with their experiments. So currently, in my lab, we work with a variety of different experimental models. The work that I show you today is all done in Arabidopsis plants, which you've already heard quite a lot about over the last few days. We're also working with a couple of different crop species, and we're working with some non-photosynthetic organisms as well. We have a collaboration on the malaria parasite, for example, at present. So let's look a little bit about how circadian rhythms, how those 24-hour cycles get generated inside cells. So one way of thinking of the circadian system is like a series of signaling pathways that process environmental information. And at the heart of this is the circadian clock, or what we call the circadian oscillator. This receives information from the environment about the presence of light when it's daytime and about temperature fluctuations. And that information about the environment gets communicated to the circadian clock in order to align its timing with the timing in the environment. The circadian clock produces this 24-hour biological cycle inside the cell, and that information about the time of day that comes from the clock is communicated through output processes to, at least in plants, to operate primarily by regulating gene expression. So some genes get turned on at certain times of day, and other genes get switched off, and then that changes across the day. So different subsets of genes are expressed at different times of day. The way in which this circadian oscillator produces a measure of the time of day fascinates a lot of people within the field. And I'll try to explain to you a little bit about how it works, how we think it works. So most, but not all, circadian clocks that are found across life are transcription, translation, feedback loops. So they're genes that are connected to each other and regulate each other's expression. Here is an example of this. This is a made-up example. It's not a real one. So let's say we have a simple biological oscillator that is formed from two genes, gene A and gene B. So gene A, when it's transcribed and then translated, it produces a protein that we will call protein A. Now, protein A is a transcription regulator that is required to turn on gene B. So gene B is not expressed until this protein A accumulates. However, protein B that comes from gene B suppresses the expression of gene A. So what this, you have to kind of think it through in your head, but what this means is that as this protein A accumulates, gene B gets switched on, but when gene B gets switched on, protein B accumulates and turns off gene A, which ultimately leads to gene B being switched off. So it goes round in a cycle. And that forms a simple biological oscillator. There are other types of this. If this has the right kind of biochemical rate constants in it, it can produce a 24-hour cycle. As ever in biology, things are far more complicated than this. And the plant circadian oscillator that we've learnt about mainly through the study of Arabidopsis is infamously complicated. So don't worry about this next slide too much. It's mainly to show you that it's complicated. So what these are in this diagram, these names of things are the names of all the different genes and proteins that are thought to form the kind of core of the Arabidopsis circadian rhythm generating system. You don't need to remember the names of the genes, but I'd like you to notice is how interconnected they all are. They seem to be formed into a few different loops, like there's a loop over here, there's a kind of loop here, and then there's a loop here as well. And these are sets of genes that are expressed at different times of day. And then other things that you maybe can notice are that there are arrows on some of the lines and there are kind of blocks on some of the other lines. So some of the steps within the functioning of this network are activation steps and some of the steps are suppression steps. It's hard to imagine in your head how this might fit together and produce a 24-hour rhythm. So some labs that study how this works have constructed mathematical models that contain all of these components and the rules about which one regulates which one and whether it makes it go up or down. And by doing this, you can kind of formulate mathematical models that help you understand, oh, well, if I took out this gene here, how would it change the functioning of the clock? So it's kind of a non-intuitive network. The complexity of the network is thought in itself to provide robustness to environmental fluctuations, because there are so many interlocking components. Another thing to notice about this is basically different parts of this network are expressed at different times of day. So some genes are switched on in the morning, some genes are switched on at the end of the day. And the morning kind of begins with the expression of these genes called LHY and CCA1, and as the day progresses, different genes become expressed. And so this kind of cycle with this complex network is what sits at the heart of the circadian clock. It's thought that in Arabidopsis at least, each cell has its own semi-autonomous circadian clock, and that contrasts in mammals where the circadian clock is in the suprachiasmatic nucleus of the brain and then produces timing signals that go through your body to synchronize sub-clocks. So plants are very decentralized. They have lots of leaves, they have lots of roots, and that decentralization extends to molecular processes such as the circadian clock, where each cell has its own, seems to have its own clock. In plants, the circadian clock controls tons of different things, and I tried to summarize this on this diagram. So around the morning, the circadian clock drives the opening of the stomatal pores within the leaves for the exchange of gas during photosynthesis during the day. And the clock also controls photosynthesis inside chloroplasts. As the day goes on, starch accumulates, and the regulation of starch accumulation is controlled by circadian rhythms. And then at night, the stomatal pores close, and in some plants, such as legumes, the leaflets fold up. And there's a lot of growth occurs at night through starch mobilization, and the clock also very carefully times the use of starch reserves so that they only run out just around dawn, and perturbations to the circadian rhythm cause plants to starve because they mistime the use of their starch during the night. There's other, as well as these kind of folding patterns of the leaves, there's some other very obvious types of circadian rhythms that happen in plants, and these are leaves of a succulent plant called Mesembryanthemum crystallinum, and you'll see the flowers open and close with a circadian rhythm. And while we're on the topic of flowering, as well as controlling scent production and floral tracking, solar tracking by flowers, the clock also is involved in the measurement of the seasonal changes in photoperiod that determine the season when some plants flower, such as cereals, which produce a lot of our food. So circadian rhythms are kind of very, very much embedded within the whole life history of a lot of plant species. And at least in Arabidopsis, around 30% of the genes on the genome are under circadian clock control. And the numbers seem to be similarly high for a lot of other plant species. So the clock is really integrated in everything the plant does. But that's perhaps not surprising when you remember that plants need light to grow. And if they can anticipate when it will be light and when it will be dark, you could see that it will provide a benefit, really. The first piece of data I'd like to show you, some experimental results, was something that I did quite a long time ago when I was a postdoctoral researcher. And this kind of acts as a nice example of how valuable circadian timing is to plants. And I'll talk you through these two graphs. So have a look at this graph on the left first, please. And what this is showing you on the y-axis is the amount of carbon being fixed by photosynthesis by Arabidopsis plants. And this experiment compares two different strains of Arabidopsis. This one on the far left, which is called Col Nort, is the wild type plants, and they have a normal functioning circadian rhythm. And in this experiment, their photosynthesis is compared with some plants where the circadian clock has been stopped by overexpressing one of the genes that forms part of the clock. So these plants lack a circadian rhythm, and they also fix less carbon through photosynthesis. So a loss of correct biological timing seems to reduce carbon fixation. Have a look at this graph on the right as well. This is the amount of biomass accumulated in the leaves of Arabidopsis plants that were grown for 4 weeks. And the 3 bars on the graph are all from wild-type plants with a normal circadian clock, but they're grown under 3 different conditions. The central bar, where it says T24 at the bottom, is plants that were grown under a 24-hour day with 12 hours light and 12 hours dark. And then in the other 2 bars, the plants were grown under slightly different conditions. So under T20, it's a 20-hour day with 10 hours light and 10 hours dark, so it doesn't match how the day-night cycle is on the Earth. And T28 is correspondingly a longer daily cycle. And what happens under these kind of long or short days is that the process of biological timing in the plant becomes misaligned from the daily cycle in the environment, because the period of the internal clock doesn't match the period of the environment. And what this does is it causes the plants to mismanage their starch reserves, and so you get they grow less well. They accumulate about half the biomass. So this is kind of showing you on the left that having a functioning circadian clock is important for plants, and on the right, having an accurately timed circadian clock is important for plants. So something that I alluded to earlier was that the circadian clock has some kind of control over photosynthesis. And something we've been interested in my group for a number of years in is the signaling processes that communicate the time of day from the clock, which is within the nucleus, and like, how does that information get into chloroplasts to control rhythms of photosynthesis, or to control daily cycles of photosynthesis. And I'll just show you 1 or 2 slides of results related to this, where we've uncovered some interesting things. But I have to give credit to all the people in the group that have contributed to this line of work within the lab, and some fabulous collaborators as well, shown down here on the right, Tanaka-sensei and Hanaoka-sensei at Tokyo Institute of Technology and Chiba University respectively, and Heather Knight in Durham as well have been great collaborators on this project. So we wondered how information about the time of day is getting into chloroplasts. And so the thing that you need to know to understand what we did is that chloroplasts have their own genome, right? So as well as the genome in the nucleus, chloroplasts have a small genome that is thought to derive from their ancestry as cyanobacteria that were engulfed by cells that eventually became plant cells. So they have a small circular genome, it's separate from the genome in the nucleus, and it has bacteria-like processes of gene expression rather than eukaryote-like processes of gene expression. And the genes encoded within the chloroplast encode essential components of the photosynthetic apparatus. So if they're not expressed properly, the plants don't photosynthesize correctly. Something we noticed is that a proportion of the genes in chloroplasts have rhythms in their expression, and some of them reach a peak expression in the daytime, and some of them have a peak expression in the nighttime. And we think that about 70% of gene transcripts in chloroplasts can have a rhythm, if you look at all the experiments that have been done to check this and put all the results together. So we wondered what might be allowing the clock within the nucleus to regulate these rhythms inside chloroplasts. And to understand this, you need to know a little bit about how chloroplast genes are expressed. So in this diagram, this blue circle's the nucleus, and the green rectangle is a chloroplast. And chloroplast genes divide into two categories that rely on two different mechanisms of expression. And one is regulated by something called nuclear-encoded plastid RNA polymerase. It's a protein that comes from the nucleus, goes into chloroplasts, and regulates the expression of one set of genes. There's another kind of gene expression system. This is the same as the bacterial E. coli gene expression system. It's called plastid-encoded plastid RNA polymerase, and it's encoded within the chloroplast. So it's a multi-subunit protein that comes from the chloroplast genome, but it requires also what we call a sigma factor, which is encoded by the nucleus and is required for transcription of the genes to start. So sigma factors, they're prokaryotic RNA polymerases that sometime during the history of plant evolution moved to the nuclear genome. Now, Arabidopsis has 6 of them, and they provide a kind of network that allows the nucleus to control which genes are expressed inside chloroplasts. Coming back to the clock, we noticed that 2 of these sigma factors have circadian rhythms in their expression. And we thought, well, let's wonder whether these are controlling rhythms of expression inside the chloroplast genome. So I'll show you an experiment where we tested this idea. So let me talk you through this graph. So what this graph is showing here, that's on the left, on the x-axis is the amount of time that the plants were under constant light conditions. If you remember back to my introduction about circadian rhythms, I said you detect circadian rhythms under constant conditions because they're generated from inside the cell. So the plants were under constant light conditions, and the y-axis on the graph is the level of expression of the mRNA for this particular sigma factor that we chose to use to test our question. Now, the yellow line on this graph is wild-type plants, and the expression is high at certain times of day, and it's low at other times of day, and that pattern repeats with a 24-hour cycle. So now, because of what I've told you earlier, you would know that has a circadian rhythm in the expression of the transcript. The blue and the red lines down here are 2 mutants that don't express this gene called sigma-5. So basically the signal flatlines in the expression because it's not expressed. So then when we asked then in our experiment, well, what happens to the chloroplast genes that this is involved in controlling? And so we did that experiment. This is a chloroplast gene. Don't worry too much about what the name of the gene is, but we use it as a reporter to measure levels of chloroplast gene expression. And again, this is the same kind of experiment where the x-axis is constant light conditions, and the yellow line is the wild-type plants. And I hope you can see there's a circadian rhythm of expression there because we've got the repeating 24-hour cycle in the pattern of gene expression. And the circadian rhythm is abolished in the 2 mutants of the nuclear-encoded gene sigma-5. So this suggests that this gene within the nucleus is controlling rhythms of expression of this gene inside chloroplasts. We had to test this in a variety of other ways as well for the publication. So one of the experiments that was really crucial was that we changed the timing of expression of this gene. So it was expressed during the subjective night or during the night rather than during the day. And it resulted in a corresponding change in the timing of the expression of this gene. And so that, in combination with a few other things, led us to conclude that this is communicating timing information from the nucleus into chloroplasts. So there were a few big ideas that came out of this study that we did. So the first of these is that circadian timing information can be communicated between different organelles inside a cell. And I think that idea holds, you know, that we tested this in plants, but you could equally think, oh, you could test it in fungi or in animals or something like that. So it's kind of tackled a bigger question. And something that I think you as potential plant biologists, I'd like you to take away from it is that we discovered this for the first time in any organism, but we were using plants as a model to make this discovery. And this kind of illustrates that plant sciences as a field has a real power to obtain general insights into biological systems that go beyond just what we might find out about plants or their benefits to us, okay? So this is a general insight that goes across all of life rather than one that is restricted to knowledge of just plants. So it's kind of very important to know that you can do this with plants quite easily and quickly compared with some other types of experimental models. I'd like now to show you a second snapshot of research that we've been doing in the group. This is a little bit more agriculturally facing, and kind of like to put this question to you about whether knowledge of circadian rhythms could be used to improve the efficiency of agriculture. And I'm going to talk you through something that I think might make some of you angry, and that in part is deliberate, because it might lead to some discussion, or you might think, or it just might make you think, you know, and that's the idea really of this week. So this is work actually that was led in my group by an excellent former PhD student, Fiona Belbin, and we did this work in collaboration with Kerry Franklin at the University of Bristol, and also a couple of good collaborators at a company called Syngenta, which is based near to Reading in Berkshire. They have a good research campus. The thing to remember here is that by working with Syngenta, we were able to do something that we wouldn't have otherwise been able to do because they have specific aspects of knowledge to bring to the project. So sometimes working with companies can be quite helpful, whereas other times it is not always as helpful. So let's go into this story. Something that's been known for quite a long time is that there's daily variation in how effective some herbicides are. Herbicides are important for agriculture for a variety of reasons. You know, they increase the crop productivity, they allow prevention of movement of weeds into areas where crops are grown. You sometimes see that just the edges of fields get sprayed to do that. And under some circumstances, they're also used to kill the top growth of plants before harvest so that they can be harvested more consistently. And so it's been known for a while that the effectiveness depends on what time you spray. And this is from an old paper, it's not from my group, where they applied the herbicide called glyphosate at different times of day and found that they got much better weed control at certain times compared with other times. And when we saw this, I thought, oh, this looks interesting because, you know, it could be you see these kind of phenomena because there are daily changes in the position of leaves or daily fluctuations in the humidity in the air or whatever. But you know, I wondered if there could be a biological explanation for this, because understanding it a bit better, we reasoned it might enable us to use agrochemicals perhaps more efficiently, like reducing the amount of time that farmers have to spend spraying, or potentially reduce the amount of the chemical used, so limiting environmental damage and reducing cost. And we wondered if the circadian clock might underlie some of these cycles in responses to herbicides. And so tried to think about a way to test this, and I got talking to the people over at Syngenta, and we thought, well, let's test this using glyphosate because it's really widely used. It's, you know, a very large amount of money is spent on it worldwide. It wasn't going to cause us a problem to work on it because the patents are all expired now, so there's there's not really the kind of trade secrets within the companies in its use anymore. And the way it works is very well understood. What it does is it blocks in plants, there is a biochemical pathway called the shikimate pathway that is not found in mammals and other organisms. And it blocks an enzyme called EPSP synthase that is at an early stage in the pathway. And this prevents plants from synthesizing aromatic amino acids as efficiently. And my former student, Fiona, started thinking about this, and she wondered, well, how could we test if there is a circadian rhythm in the sensitivity of plants to glyphosate? And what she did was she looked at papers that had published studies showing how the genome in plants responds to glyphosate. And she also looked at studies showing how the genome is clock controlled. And she looked for common genes that are regulated by both processes. And what she noticed was that this set of genes was enriched in auxin signaling related genes. And the reason we thought this might be the case is because some aromatic amino acids are used as precursors for the synthesis of auxins within plants. And so Fiona devised a simple way to test in the lab this more general question about glyphosate. So what we knew is it blocks a metabolic pathway that's needed for auxin production. And we thought, well, what is something very convenient and simple and straightforward that we can use to study effects of agrochemicals on auxin signaling? And we used Arabidopsis for this work. And so what Fiona did was she decided to measure the elongation of the hypocotyl, which is like the stem that comes out of seedlings. And this stem, this hypocotyl, elongates through cell expansion when seedlings emerge. And that cell expansion is cell expansion rather than the process of cell division. And it's regulated by auxin signals. So this provided us with a tool to experimentally ask questions about whether there might be rhythms in the effects of glyphosate. So Fiona went off and tested this, and she came back with these data, and I'll talk you through these. So this top slide, this top graph, is an experiment where Fiona took separate batches of elongating seedlings and added a glyphosate-containing herbicide at different times of day. And she measured how much the elongation of the hypocotyls was reduced by adding glyphosate. So when the bar is higher on the graph, the elongation of the hypocotyls was blocked more, whereas when the bar is lower on the graph, the hypocotyls grew more freely. And what Fiona noticed was that when she applied, under constant light conditions, when she applied glyphosate around dawn, it caused a greater inhibition of hypocotyl elongation compared with later in the day. Fiona wondered whether this was due to circadian rhythms, and so she took an Arabidopsis line where the clock had been stopped by overexpressing the CCA1 gene, and what she found was that the daily oscillation in sensitivity to glyphosate was abolished. So we think that at least this response under these conditions in Arabidopsis was being controlled by the circadian clock. So Fiona then went on and she said, well, if plants are more sensitive to glyphosate around dawn than around dusk, how much more glyphosate do I need at dusk to create the same block on hypocotyl elongation? So what she found was that towards the end of the day, she needed like 2.5 to 3 times more glyphosate to have the same effect as around dawn. And that difference was abolished in plants where the circadian clock had been stopped. So the way that we interpreted this was that the clock underlies a rhythm in the effective dose of glyphosate. The interesting thing about this is that, so this was done in the lab under controlled conditions using an experimental model plant, Arabidopsis. But if this idea or this concept scaled to crops or things grown outside, you could imagine that it might be possible to tune the concentration of the agrochemical used according to the time of day in order to reduce the amount that you have to put onto plants. So what we found through this work is that the circadian clock controls a response of plants to the most widely used herbicide. But this opens a bigger question, really, of so this was the first evidence of interactions between circadian rhythms and anything that's used in agriculture. We termed this idea agricultural chronotherapy, which is reminiscent to the situation in pharmacology and medicine, where drugs applied at certain times of day can be more effective than when they're applied at other times of day. You could also possibly call it chronotoxicity, because herbicides are toxins. I mean, it's possible that this idea might scale to other kinds of things like herbicides that are added onto plants, or biostimulants, or other things that are added into fields, and there might be fluctuations in responses to them. So this was a kind of, the thing that I'd like you to emphasize to you is it was a proof of concept study rather than a practical application study. But it also demonstrates a new general principle in biology that could apply across the use of agrochemicals, and it kind of opens a lot of questions that people could explore. So I'm at the end of my talk, and thank you for listening. So I think that the take-home messages I would like you to go away with, and we can have a discussion about it afterwards, are that many kinds of living organisms can tell the time, including plants, and these rhythms are really important for plant productivity. And the other point that I would like you to take away is that plants represent a really good experimental platform to explore more general ideas in the biological sciences and get insights that go right across life rather than are just restricted to plants. I need to thank my fantastic research group at the John Innes Centre, because they're the ones that have contributed to all of this, and all the former members of the group, and my collaborators and funders as well. Thank you so much for your kind attention.